Hello, everybody. My name's Tim Julian. I'm at AVOG. It's a water research institute in Zurich, Switzerland. I just started a month ago. And I have a confession. I've never worked with leptospirosis before. But I think I was invited here to talk about some of my microbial exposure work. So I'm starting at AVOG a group devoted to microbial exposure in human health, with the goal of studying pathogen transmission at the boundary between people and the environment. So I'm going to give a commercial here. If anybody's interested in learning more about AVOG or coming to work, at AVOG we have some funding sources for short stints, and I'm also looking to recruit PhD students and postdocs in the next couple of months. So what my group is really going to be focused on is looking at the concept of beta, the movement of people from the S compartment to the I compartment in the SIR models that we've seen a lot about, as a function specifically of the interface between people and their environment, that microscale period where it's a person's direct interactions. And how do we model that? How do we understand that? Factors that influence pathogen fate and transport. And Lorenzo gave a great introduction to diarrheal diseases. That's my main focus. So I won't go into too much detail, but I work with viral gastroenteritis, bacterial gastroenteritis. Those are the main pathogens of interest that I've worked with. And I also work with fecal indicator bacteria, and that's going to be the output of some of my studies. But what I wanted to talk about first was how do we model our microscales, and what do I mean by microscale interactions with our environment. And so some of the work I started with was modeling a really simple interaction of a child with their individual toy. So it's a really simple model. And the way I modeled it is first you start with the toy or the surface, and then the child has two hands that are interacting with that surface, and then they can become infected through contact of any of those surfaces with some sort of a membrane. So if we think about leptospirosis, the facial membrane dose that I'm using here for rotavirus is relevant, but perhaps other concepts of how you would become sick and dose might be an open sore in addition to the oral route. In addition, in these models, you often have to account for changes of the pathogens within the environment. So in my model, I accounted for inactivation of the virus over time on these surfaces. And so the way that I modeled this specific scenario is as a discrete time stochastic model. It's a mechanistic stochastic model, so it looks at tracing the actions as they happen and translating that into to exposures and dose. So for here, as an example of the dose from hand contacts, so a child's dose of viral gastroenteritis through contacts with their hands. So you have to account for various different factors in order to understand the mechanistically what's happening. Rotavirus on the child's hand has to move into their mouth through a number of different factors you have to account for, including the transfer efficiency, how much of the virus is transferred when there's contact. The surface area, and here in the model I use the total hand surface area and then the fraction of the hand that comes into contact with the mouth. And then I have the concentration on the hand, and then you account for inactivation. So how do we understand hand contamination? Oh, go ahead.
That's one question. Not taking into account here the frequency of contact between hand and...
I'll get to that.
Oh, I'm sorry.
Yeah, that's great.
Is this like a rate in one process?
I'll get to that, too. Great questions. So how do we model hand contamination? It's similar to how we model dose, where we have concentration on the hand as a function of the previous hand concentration of the virus on the hand, always accounting for inactivation, and then transfer between the hand. And in this case, the hand is contacting the fomite. So you have hand concentration, fomite concentration, accounting for inactivation over the time on each. The difference between the concentrations, because transfer is modeled here as a function of the difference, and then the transfer efficiency, the percent of that transfers. So for each one of those parameters, the transfer efficiency, the inactivation rate, the initial contamination on the of the virus on the surface, they're all modeled as distributions. And this is where the stochasticity comes into play, where every time that the model is run, it iteratively chooses a point within that distribution and uses that to continue the model. And then the model is run, simulated thousands of times, and we end up with a distribution of outcomes. And so
you draw from the distribution at each time point within a single run.
Mm-hmm.
Right, so it's an ergodic process rather than.
Mm-hmm. So here the ones that I modeled for this specific scenario include the initial virus concentration on the fomite, the area that's contacted each time that their contact has occurred, the fraction that's transferred on each contact, the inactivation rate, which is modeled for the whole process, and then activity data. And now what do I mean by activity data? I mean the frequency of hand contacts. So how do we know this? How do we get at this information? And there's a lot of different ways, and I think this is really relevant to this community in particular. One of the cheapest ways and the least accurate ways is to just ask, how many times did you put your hand in your mouth in the last 4 hours? Well, who knows? So another way to do that is to have somebody standing in the corner watching you, or in this case watching the child, and counting every single time. That gets a little bit more accurate, but a little less reliable. And so one of the ways that we work a lot with is we actually videotape people in their environments interacting with their environment. And then we go through a process of recording every single object that whatever the body part of interest contacts over the whole duration on a second-by-second basis. This is known as micro-level activity time series. So in this scenario, you have a child playing with a toy, and their right hand comes into contact. You track the right hand contacts with every single surface in their environment over time. And you record, and I'll get a little bit more into detail about exactly how this works. But you end up with this micro-level activity time series, which shows that for 10 seconds, the child's right hand was contacting an object that was classified as a metallic object. And then their hand went directly into their mouth, and then they dropped everything. They weren't touching anything. And then, again, they picked up whatever that toy is. And so this is a sequence of events. In addition to the sequence of events and the duration of the events, you can rewatch the videotapes if you're patient enough and classify the fraction of their hand that's contacting each object and the number of fingers. And then you can also classify what they're doing on a macro activity timescale. This is obviously very expensive and laborious. So now what are the outputs of our models? So this is one example of one simulation of the exposure for their right hand and left hand and fomite concentrations. So what you end up with is the child's interacting with the toy over time. You can model the concentration. And what we see here, and one of the major findings of this model, is that a child only has to contact a contaminated object a couple of times for the majority of the contamination on a per centimeter square basis to transfer to their hands. It's only the first couple of objects before the fomite has contaminated their hands about as much as it will. And now you also can see that this decreases slightly over time due to removal of viruses through dose and through inactivation rates. So now I'm going to take a little bit of pause and say, so we have the modeled concentration on their hands. We know the frequency of mouth contacts. What we're also getting at is from those mouth contacts we get at, that's dose. That's the amount of virus, in this case rotavirus, that's gone into their system. Now how does that translate into risk? So one of the frameworks for this are dose response curves, which we've talked a lot about today. And so I'm going to talk first about the rotavirus dose response. These are the fraction of whatever the subject, in this case it's people, so I believe it was grad students who were recruited for this specific data set. The fraction of those students who consumed this amount of dose that became sick as a result. So here, for example, this is one rotaviral particle. And about 10% of the people, which is probably 1 out of 10 in this case because the sample sizes are relatively small, and then it increases as you increase the mean dose. And then what you can do, Peter Tunis did this in 2009, is he fit a series of different models to that data. And from that, you can estimate the probability of risk. So for example, if you consume 10 rotavirus particles, that corresponds to, I think, a 62% risk, roughly. So this data translates that dose model into risk. Now, there's a number of drawbacks specifically for my system, which is that this is adult grad students, and I'm modeling a child. And so you can develop parameters, or you can just kind of use this information. So what you end up with is, after you complete the simulation, as Matteo mentioned, you can deconstruct this model into its factors using sensitivity analysis and decrease the complexity. So we had this very complicated system of frequent contacts with stochastic processes. And what we find is that the major driver of risk of illness is the initial contamination on the fomite that the child's interacting with. And the reason is that there's so much uncertainty about how much contamination is on the object that the child's contacting. So we modeled it from 10 to the minus third log 10 virus per centimeter squared, all the way up to 10 virus per centimeter squared. And what we found is that the vast majority in risk of illness variability, if you imagine just a one-dimensional graph, you see a huge range between 0 and 80% risk of illness over the whole simulation. And when you deconstruct that by initial concentration on the virus, you reduced your variability within the system to roughly 10 or 20% risk. So that's just an example of the output of our models.
Is that for these simulated child-playing toy type scenarios?
Mm-hmm.
So it's a relatively specified type of contact, having to integrate over all kinds of different natures of contact?
It's exactly that. It's this very specific confined system. But a similar story can be told in other systems. And I'll talk a little bit more about that. So I'm going to take a pause and switch over to the leptospirosis dose modeling. And this was inserted as a direct result of yesterday's conversations. I came across two papers. One came out in risk analysis really frequently that I just put up here to show people that there's some data on this. And maybe some of you have seen this before. It's just showing the dose response model for leptospirosis for I think it's this specifically is 8 or 9 data sets, or maybe fewer than that. And you can just shows over time the model, the probability of mortality for the animal. These are animal models as a function of the dose changes, obviously with increasing number of days. So you start with 9 days. There's a greater likelihood that the animal will still be alive, but then progressively the animal decreases. So this paper is interesting in that it models leptospirosis probability of infection. But an interesting finding from this paper is that all of the studies they reviewed, they were all subcutaneous or interperitoneal infections. The second thing that I want to talk about is this paper that came out in 2009 that talks about chronic exposures, multiple chronic exposures, as opposed to one acute infection. And so I'll just walk you through it really quick. It's something that you might want to check out. So there's so the probability of infection for this is Campylobacter, I think, is what the model is for this. So the risk of infection from 300 colony-forming units is roughly 75% if you're exposed to it once. And this model that they used to look at multiple exposures, in this one, there's 4 inoculation events that sum to the same dose. So it's 300 colony-forming units. But it's sporadically over time in evenly distributed chunks. So it's 75, then 75, then 75, then 75. And what they show is here, your probability of infection has dropped now to 45%, even though it's the same dose. This is how they choose to model it. Now, under a different set of parameters, these are 2 different parameters to understand the dose response curve. It might, in fact, only drop to 65%. And then the only reason I'm showing this to you is to show you that the mathematical model exists for these chronic exposures. we also have a mathematical model to explain acute exposures for leptospirosis. So it's clear to me that if we want to understand chronic exposures to leptospirosis, we have the mathematical framework for doing that. It's just a matter of lining that up with biological data to validate.
Was this informed by biological data?
Yes.
So they did the experiments in...
Oh, no. No, it was informed, I believe, by acute exposure data, so not by chronic, but I'm not exactly sure.
One time,
Yeah.
so they informed that model by the people who were infected with 75, and the new people who were infected with 30?
I don't know. I'd have to look at the paper more in depth. I just looked at it cursory, very briefly. But I don't think there are very many studies at all that have done the repeated exposures with biological data.
I mean, the one that I think is a famous one is the tuberculosis, right, where they put guinea pigs in hospitals and trying to help school. They looked at short exposures versus long exposures in TB wards.
Oh, interesting.
And that's where the mathematical model goes driving.
Okay. Cool. So back to what I've been working on. I'm just going to go through really quickly a case study of Peri-Urban transmission in Tanzania. This is the F diagram. I don't know if any of you are familiar. It's just a way of modeling transmission from an infected host in human-to-human transmission through the environment. And I'll just skip ahead to some stuff. So the traditional model in peri-urban Tanzania is that stored water quality in the home is really important. And so the primary recommendations are you should be treating your drinking water. And so there's some studies that have shown that, in fact, the water that you collect at the source, so your borehole, is clean, and then as you transport it home and as it stays in your household, it becomes recontaminated. And so further studies that are looking at why is this water being contaminated with bacterial and viral pathogens in the home linked it to recontamination from just hand contact with the soil, or with the water. So frequent water contacts with hands were increasing the presence of fecal indicator bacteria and pathogens. And so we're suggesting that maybe that in addition to the intervention of treating the water, which is the box on the right, there might be interventions like hand washing to reduce transmission of pathogens into the stored water in the first place. But then we're also wondering how are these pathogens, fecal indicator bacteria and pathogens, getting on the fingers in the first place? And so we did a study where we looked at hand contamination. What we found is that in these settings in peri-urban Tanzania, there were thousands of E. coli per 2 hands, and there were thousands of enterococci per 2 hands. And these are really high levels. In the United States, we conducted a similar study, and the vast majority of hands had less than 10 E. coli. So these are dramatic differences between settings. Yeah. So we took our model of trying to understand human activities with their environment to understand how hands are being contaminated. And we did this by looking, by sampling hands for fecal indicator bacteria, asking the women to wash their hands as they normally would, collecting a second hand sample, and then asking them to perform one activity, their typical activities as they would normally perform in their house, sweeping, preparing food. And then we collected the third hand sample. And we looked at the change in hand contamination as a function of activity. And so sitting was our control, just sitting there. And we saw a decrease in E. coli and then an increase in enterococci that was not statistically different from 0. But for all other activities, concentrations jumped up dramatically. And this included bathing, cleaning dishes, preparing food, and sweeping was a really big one. And we were wondering, why is it that any activity these women do besides just sitting there is increasing their hand contamination? So we took the framework for our model of the child interacting with their environment and applied it to 2 women in a small pilot study. And so we shifted the focus of the camera to a first-person perspective or ego perspective so that we could track the contacts, and then we use this software. This is how we translate, how we collect the micro-level activity data. It's called video track software, and it's every single object that you identify by watching the video at one time that the woman comes into contact, their location, and then whether it's a constant or a repetitive contact. And we translated that into the micro-level activities time series. So then again, we defined the model parameters. And this, we used a deterministic model for this as opposed to stochastic. We collected small amount of information on concentration of fecal indicator bacteria for each of the surfaces that were contacted. Very little information. This is very sparse data. And then we also looked at transfer percent, which we collected from the literature. There's a number of studies that have looked at bacterial transfer between hands and different types of surfaces. And then we modeled using this information approximations of the right hand contamination. And this was done, the initial hand contamination as well as some of the surface contamination was modeled using microbial sampling data. And so this is an example of that exposure output where you have cumulative over time concentrations on their hands, on the women's hands, and then the measured contamination level. And then it was validated with these data points. And so what we see from our model is that contact with soil and sand happened once, but the contamination was so high that the concentration shot up to, or it happened twice. There's 2 rapid succession contacts with soil. And then, over a period of time, as the woman was washing her clothes, we modeled that the hand contamination would decrease down to nothing, which was validated by that data point. So clearly, there's sparse validation data here. So we don't know if this data point is true. But the suggestion from that one soil sample we collected was that soil contact is driving contamination. So in addition to that potential finding that I'm going to talk a little bit more about pursuing in a second, is the power of this type of an exposure model is that you can look and investigate how different factors might influence different curves. So here, water contamination was modeled as 25 colony-forming units per 100 mil, the water that was used for washing clothes. But if you change the different factors by decreasing or increasing contamination, you can see how it impacts the exposure curve. So here, for example, decreasing the contamination in the water used for washing won't change the hand contamination, but increasing will dramatically. Also, you see here the validation points didn't line up with the exposure model, which suggests that maybe there's an issue with the model. So following the finding that soil could be a driver of contamination, we looked at a number of different points throughout the house, and we found really, really high concentrations of fecal contamination in the soil throughout the whole house. And we originally thought that the latrine might be the source of the fecal contamination, but in fact, fecal contamination was lower at the latrine than it was at or in the house or where food is prepared. We also dug down to 10 centimeters at the latrine and at the house, and we showed dramatically reduced contamination of fecal contamination below 10 centimeters. And then the fecal indicator bacteria coincided with the presence of pathogens, specifically enterovirus and rotavirus were found at the house entrance along with 5 different diarrheagenic E. coli. So I just threw this in to show one of the other projects we're working right now on is urine collection. And so this is an example of this model framework applied to urine concentration, where dose is a function of the skin-mouth contacts, and then the transfer efficiency of a pathogen from urine to skin, which is there's no data on transfer of pathogens between a liquid and skin surface. This is poor resolution, but it shows that that percent transfer will really drive the ingested dosed risk curve. So the overall summary of my talk is that boundary conditions really matter. You need a lot of information for these exposure models, including pathogen characteristics, human interactions with the environment, and then there's a lot of limitations still. But I think these models are very valuable. So just like to thank everyone at Stanford and Johns Hopkins and also AVOG where this work was done. So thank you.